How's everybody doing? Last session, and everybody's looking happy still. So that's a good thing. So we're going to get the spatial session started. First, I want to thank ABRF Organizing Committee for giving us a chance to come up here and talk to you about our experiences with spatial. I also especially want to thank vendors like NanoString for helping us fund this meeting so we can all get together. We're going to start with our corporate sponsor talk by Prajan. And then we're moving on to Katie Petentler, who has been at Stowers doing spatial work. Her abstract was selected for a lightning talk, so I'm excited about that. And then we'll introduce, the rest of us will introduce ourselves and move on from there.
Thanks so much. Pleasure to be here. So pleasure to be here. On a personal note, this is my first time here. I've been with the company for 5 years. And I think this conference in particular, I really appreciate because the same way that we're technology developers, many of you are the providers of the technology. And I think that's something we share in common. So I've really enjoyed the conversations we've had. Great to see a lot of familiar faces we've worked with over the years. So with that, we'll get started. So before I get started, I titled this Sample to Insight Journeys with Highplex Spatial Data. And the most important letter in this title is actually the S at the end of journey. And the reason I call it journeys is because I think one thing we've learned throughout the rise of spatial is that it's really an interdisciplinary, cross-functional exercise to really get these spatial technologies to get the most value out of it scientifically. And what that really means is there's many independent parties, whether it's in the technology development side, the service provider side, the computational biology side, the bench scientist side. We're all going on these journeys independently and then meeting together and making sure we're consolidating. So what I want to do in this talk, hopefully, is give you a sense of the different journeys that exist for the different players and how we've been innovating along those lines. So with that, we'll get started. So the very first journey that's very exciting to talk about is our journey as NanoString, the spatial journey we've taken starting from 2019. I joined the company at the start of 2019, and it's been amazing. I mean, at the time, we had the Genomics spatial platform, and we were profiling in space 80 RNA targets. At the time, that was about 10 times more than anything commercially. And from there, we went from 84 to 1,800 to every protein coding gene virtually. And then we went back and did that all over again, except at the single cell subcellular resolution. So it's been a really, really fun journey. And it's been a vision driven around plex, resolution, and throughput. And we're going to talk about all of that. And it's been driven by feedback from all of you and the needs for what we need in science. So there's 4 green boxes on the slide you'll see, and these are the 4 boxes I think I would like to highlight and for you all to maybe take a mental note of. One is our IPA 570-plex protein panel. One is how much progress people have already made with our 1,000-plex panel to date, the 6,000 and the whole transcriptome that's coming in 2025. So with that, we'll get started. So what we've seen is that if we think about spatial biology platforms generally, we've seen that we can break them up into generally 2 categories. Profilers and imagers. Profilers tend to be focused more on throughput and focusing on tissue structures, while if you think about imagers, they're really focused around resolution, subcellular resolution. And there are some trade-offs and benefits, and ultimately we see them as complementing each other. Generally, profilers have a much higher throughput and plex, but what you can get with the imagers, of course, is that high resolution. You can start getting into single-cell and subcellular interactions. So what does this mean visually? Without getting into the exact technology, I'll assume many of you have a general idea how these technologies work. I know Bony mentioned, I was just talking to him a couple of days ago, he mentioned seeing is believing. So this image here will describe to you what the 2 different technologies do. So if we look at the pancreas, you can just look at some of the islets in the pancreas. You can see here with the Genomics, what you can do is ultimately select cell populations. So we can look at alpha cell populations, beta cell populations, and you can profile the whole transcriptome. And if you think about Cosmics, ultimately what you can do is profile those same structures in cells, but now what you're doing is actually looking at individual RNA molecules inside individual cells. So it's the idea of thinking about structurally at a higher plex versus subcellular at a lower plex. And when we keep talking about RNA, I think RNA is super, super exciting, and we're going to see a lot of great RNA work in this presentation. But our history is actually starting from the spatial protein world. And that's one of the first innovations that we made in November 2023 that we launched. So our early discoveries in genomics were all protein-based. And what you're seeing here is a back-to-back series of the first early genomics papers that were all high-impact Nature papers focused on looking at tertiary lymphoid structures, TLSs. And here on the right here, you're seeing a volcano plot with differential expression of these B cell markers. And at the time, the researchers we were working with actually questioned whether the technology was working because they weren't expecting to see these markers as something that was predicting how patients would respond to certain immunotherapies. And at the time, it turned out they didn't recognize. And as they dove deeper, they found out that these markers were actually really important. And this sort of sparked this whole, continued this revolution around TLSs and spatial biology. And what that really told us is we need to really think about expanding the opportunities people have to profile high-plex spatial. So now we have a 570-plex spatial panel that we have, which is about, I would say, 5 times higher in terms of the sheer number of protein targets you can interrogate spatially in the world right now. And what this lets you do is look at up to 570 targets and start to actually get into spatial protein discovery and not have to restrict yourself to targeted panels. But of course, I know many of you have heard, I was at the proteomics session, a lot of the discussions have been around validation and how do you trust the antibodies and what is ground truth. So definitely how we approach the IPA panel, this 570 antibody panel, is we had, of course, singleplex IHC validation from Abcam, but we also did multiplex validation by pooling all these targets together, looking at their interactions, and doing multi-tissue and multi-cell pellet array screens to make sure we're really getting the good specificity and interactions with the tissue itself. Now, what's really awesome is you can couple that with whole transcriptome that already exists for genomics to date for human and mouse. And what you can actually do is on a single tissue section, you can profile every protein coding gene, 18,000 genes, and 570 proteins on one tissue section. So this is actually quite exciting because this is getting into the realm of proteogenomics, essentially same slide, multiomics. So this is very exciting. We'll look at some of that data. And if we think about what you can do with 570 proteins, obviously you're not going to necessarily expect all those targets to show up in all your tissues. And I recognize this figure is not human readable. So if you think about every one of these bars, it's actually different tissue types. And on the y-axis, you actually have the number of proteins that are detected. So you'll see about 200 to 400 proteins being detected across a wide variety of different tissue types. So it does open up that door for you to do multi-tissue, virtually any sample tissue type. So let's look at an example where we put all this together. So this is actually a whole human embryo sample that we've done spatial proteogenomics on. So same slide, we've interrogated every protein coding gene virtually and 570 proteins. And you can look at the protein data spatially here for one target on the left, and you can see its concordant RNA expression. And then you can also take a look at when you have that discordance. I think we all know that transcription, translation, if you try to correlate RNA and protein, there's a lot of challenges there. So what's also really neat about this is now you can actually start to understand the actual concordance and correlations spatially at high plex between RNA and protein. Also in the panel, there's PTMs. We can look at post-translational modifications. You can also look at a slew of phosphoproteins spatially too, which is really, really quite exciting. And of course, if you want to dive deeper, you can move to the cosmics and start looking at single-cell interactions of those different cellular compartments. So that's what we think about. If you think about profiling, we're talking about structural profiling, multiomics, whole transcriptome with about 570 plus proteins on one slide. So then if we kind of transition into imagers, we can move to the kind of second portion of the talk, which is around single-cell spatial. And the first really exciting announcement for us was the shipping of our 6,000 plex panel. So this is the ability to essentially look at 6,000 genes at subcellular resolution on an FFPE tissue section, formalin-fixed paraffin-embedded tissue section. And what can you do with the content of 6,000 genes scientifically? So the way we designed the panel is pretty much, we call it, like you can almost think of it colloquially as the reactome panel. We designed it so that you could map the entire reactome of any tissue section you look at. And what does that let you do? So if we look at this on tissue, it's pretty remarkable every time I see it. You can pretty much, rather than projecting genes in space, you can actually project individual pathways. So what you're seeing spatially here is rather than thinking about where is PD1 or where is alpha SMA, you can actually ask yourself where is PD1 signaling happening in individual cells. And that's what you're looking at. You're actually seeing every one of these skews here refers to an individual reactome pathway that we've projected at single cell resolution across the whole tissue. So this is starting to actually bridge this idea of the limitation of RNA and lower plex RNA in terms of looking at function. And I think we've often found that we look at genes, but then we rely on proteins for function. And what's really great about having a plexity this high, pretty much 10 times higher than anything in the industry right now, is that you can actually look at gene interactions together. So it's pretty remarkable. Now, what have our users done with this already? So all of our assays, every single spatial product assay we have in the company works on FFP samples on standard microscope slides. I would say that's something that also doesn't exist anywhere else. And what's great about this is we have clinicians, for example, Dr. Will Huang, good colleague of mine, and he did his work in genomics originally, the whole transcriptome genomics work. And he wanted to extend this with the cosmics, and he wanted to look at single cell interactions around nerves and cancer, PDAC, pancreatic carcinoma. And what he did is he looked at about 6,000 genes and looked at how these genes interacted with each other. And if you think about it, a little back of the envelope math, pardon, it's a little very back of the envelope, but if you think about 6,000 genes, what that pretty much means is about 36 million directional interactions you can have of genes. And he looked at all of those interactions in an unbiased way, and he plotted those in space. And he was actually able to show single cell interactions of nerve and tumor cells for the first time in the world. And this was quite remarkable. He was able to show the interactions of nerves and tumor cells and how that affects outcome. So taking that further, I mean, I think one of the really great elements of thinking about Plex and our chemistry is it's pretty much scalable and limitless. So right now we have a pre-commercial, a whole transcriptome public data set that's available. I encourage any of you who haven't used Cosmics technology to check it out. The QR code's here, and you essentially have a public data set. This is the first ever single-cell whole transcriptome public data set for formalin-fixed paraffin-embedded tissue. So pretty exciting. And I think if I were to call out some of the key points, I would say you can find these rare cell populations that really are difficult to detect anywhere else. And you can essentially plot those on the tissue. So I think I have maybe a couple minutes left. So I want to end with some informatics, because I did say computational biology is a critical element of how we get from sample to insight. So when we think about informatics, I mean, we have our Atomics platform, which has a interactive software analysis suite. And it also has our machine learning-based cell segmentation algorithm. But another huge commitment we have, and you'll start to see in 2024 in particular, is our commitment to open source packages. So we've released multiple packages already, and we'll continue to do so. And ultimately, what we want is our open source community to utilize whatever informatics tools that benefits them. So whether it's software, whether it's open source, we ultimately want you to get the most out of this data. So we'll talk about a couple of the brief algorithms we've created to date. And if we look at what our users have done with our 1,000-plex panels to date, I would say if we just look at the instruments and we just look at how many cells those instruments are collecting before they send it up to Atomics, we are looking at around, our users have generated about 400 million cells as of yesterday. I think I pulled the numbers, about 400 million cells. And if you compare that to the Human Cell Atlas volume, that's about 6 times the throughput of the Human Cell Atlas achieved in 6 years. We achieved in about 9 months. So it's kind of exciting with these single cell technologies. They don't necessarily have the full whole transcriptome plex, but what they do have is that cellular throughput. And that cellular throughput is what's going to drive us to really look at rare cells and look at their interactions with each other. And I know all of you know this way too well, so I'm not gonna belabor this, but the idea of the workflow, how do you actually go from start to finish? And we have a workflow where you take the data, you compute on the data, you visualize, you iterate, and ultimately what you want to do is build those into some sort of standardized data formats that then you can analyze after you export it and start to leverage those open source tools. So we've tried to make the Cosmics data as accessible as we can to open source, and we'll continue to take your feedback on that and continue to build on that. And cell segmentation, we'll talk about that briefly. The most important development, I would say, is we're on probably our 4th generation of cell segmentation. It's a multi-omic cell segmentation, uses protein, uses RNA, doesn't do nuclear expansion, it uses the protein targets plus the nuclei. But what's really exciting about this is we've got a lot of feedback on the value of that algorithm, so we've added it to our informatics platform. So now any user who has Atomics can pretty much log in and work with that algorithm, and they can use the machine learning-based algorithm to tweak whatever they need. And last but not least, open source. So you ask yourself, you have 36 million spatial correlations. How are you supposed to find what is real out of that? I mean, that's a very ambitious task. So what we've created are some rapid discovery toolkits. So one of these packages is called In Situ Core. What it does is it looks at all your possible spatial correlations. It looks at the cellular locations of cell types. It subtracts that out from the data, and it actually finds for you what are the gene interactions that are actually novel, not just confounded by cells existing in close proximity, but actually genes that are interacting in proximity. So it's a very exciting algorithm. It's on GitHub. If anybody wants to check it out, just nanostring In Situ Core, and you'll see the package in the code, and please do explore it. It's very exciting. So I think I have made it somewhat briskly. So I have not kept track of time. Am I at 10? Past 10? Past 10. I'm past 10, but thanks so much for your attention. I appreciate it. Yeah.
So let's hold questions till the end because there's a lot of material to get through.
All right. Hi, everyone. My name's Katie, and I'm a member of the Sequencing and Discovery Genomics Core at Stowers Institute for Medical Research. Today, I'm going to tell you a little bit about our most used spatial assay called Seeker by Curio Biosciences, and give an example about how this has been used in-house. All right, so the Seeker kit is a commercialized version of the SlideSeq V2 technology that doesn't require any capital equipment and comes with everything you need to perform a spatial assay, including what really makes this technology possible, the Seeker tile. The tile is a 3x3 or 10x10 microarray that is densely packed in a single layer with uniquely barcoded beads. These beads are 10 microns in size, and once laid on the tile surface, they're sequenced in a way that gives not only the bead barcode information, but the XY coordinates of where every single bead is located on that tile surface. So for processing, we'll take a thin piece of fresh frozen tissue, lay it onto that tile surface, and the mRNA from the tissue will bind to the poly-DT region of the bead that's laying directly on top of. Now, since this is utilizing mRNA capture, this is really great that it's unbiased, and we can use it with the many different unique organisms that we have in our lab. After hybridization, we'll perform cDNA synthesis, create Illumina-compatible libraries, and then sequence in-house at the recommended sequencing depth. From here, we'll perform secondary analysis. That way, we'll be able to map back that cell and gene information to the intact tissue based off that known bead barcode information. Now, we had a researcher who was using the mouse colon cancer model to look at tumor microenvironments. And they were really interested to see if a treatment could change the environment and allow for cells to infiltrate. For this study, we used B6 mice that were injected with a known carcinogen to allow for tumor growth. The tumors grew for about until they were about 5 millimeters in size, then injected with an inhibitor C5 treatment. After about a week, these tumors were then dissected from the mice and used in our assay. All right, so to the left, you can see our resulting spatial transcriptomic map. We have our control sample that didn't have any treatment administered, and then our treated sample. And as expected, there is quite a difference in the spatial data looking at the cells. With our control, they kind of have like a unique and a distinct pattern with their cell arrangement. And in the treated, these cells were able to infiltrate that microenvironment, so it's a little bit more spread out throughout the sample. This data was then integrated with a single-cell data set from the lab to really get an in-depth picture of the cells within the sample. From here, the researcher then investigated this, taking it a step further, looking at specific clusters of interest to see where they were laying within that sample. Now, this was just one of the first projects that we had done with this researcher. We've then gone on to do the larger tiles to get a really nice look at the entire tumor itself, as well as a different combination of treatments to see how the cells really rearrange. This has been a fantastic tool that we've used for discovery approaches, since it is a whole transcriptome method. And we're really excited to see how this pushes not only this researcher's questions forward and allows us to get a picture before we move into a more targeted assay, say something along the lines of xenium. So with that, I just want to say thank you so much for your attention, and please feel free to ask any questions at the end of our session. Thank you.
Thank you so much. Yeah, so my chair actually told me not to give any of my opinion during my presentation. And so my responsibility is only to introduce the technology. I was just joking. So what we are going to do, I'm going to introduce you with what's there in the market, and then, and raise some questions. And the rest of the panels will actually go and dwell more detail in discussion on those things that what we learned from our experiences implementing these technologies in core facilities. So let me start with saying that this something that when we think about spatial technology, and a lot of us think like it's a new thing. So because Nature Method published this as a method of year in 2020, and then 2023 might be the year of spatial technology because so many different platforms came in the market which was actually ready for use. But if you think about really history, I'm not going to all this maze of history, but it's not something that we just started doing it in 2020 or 2023. It goes back in the early 70s when people started doing in situ hybridization. And many of you who are from developmental biology or those kind of field, you may have done some of this in your graduate studies, maybe as a postdoc, maybe as a scientist. You have done this long back. So what really changed during this year is that the ability to do multiplex and making measurements for multiple genes from the same tissue was possible because of combination of new technology, computational approaches, and then better probes or the color probes that are available for the market. So some of us like me who were actually almost in the middle of this generations when people were started thinking about multiplexing spatial platforms, we started using with probes like Stellaris and HCR, and which is now RNAscope in our studies. And now we are seeing you are looking at thousands of measurements in a given tissue. So the 2 points I would like to actually emphasize here that what we are seeing now is not something that just started in 2010 or 2020. People are using it in some or other way long back. And we got a lot of interesting data from those studies. But what now we can do, or what we can achieve now, is to make those, from single measurement, we move to 6,000 measurements or 5,000 measurements in a single tissue. And so that is a leap that we made during the last few years. So if you think about spatial technology, it can be based on imaging-based methods where you certain way you hybridize your tissue with some colored probes and then image them, and you can take multiple images and then you combine them together and then computationally resolve it for gene expression. So that is one way of doing. It's basically essentially some or other way in situ hybridization is the way that actually people are doing. Then you can also use array-based sequencing methods where you have a slide where you can actually sequence based on arrays, and then you can actually resolve the gene expression on arrays. Or you can even do sequencing, like you can see Xenium and other sequencing platforms where you are either making a rolling circle or some way you are amplifying your signal and then you are capturing that information. So that is another way. Or you can do a micro dissection. This is like a very crude way of doing it, but then it is still spatial, that you go and look under a microscope, find a very small, tiny region, and do a laser capture, and then sequencing. Because you have now low input RNA-seq method available, and that can answer your question regarding what is expressed in that particular set of cells or that particular histological landmark. So there are different ways to think about spatial analysis, and the choice of methods depends upon the question that you are asking and how much you want to spend for your experiment. Okay, so if you think about spatial technology, the key application is for dimensionality reduction and clustering. So you want to cluster your cells. What are the different cells that are actually different kinds of cells in a tissue and how they are clustering? Or you can do cell typing to know the nature of cells. You can also look at the spatially variable genes, like in a cancer or tumor context. You are looking at what's happening in the cell in the inside or core of the tumor versus outside, or those cells which are contacting the vasculature, how they are changing. So you can actually look at those interactions, cell-cell interaction and neighborhood analysis you can do. You can even build spatial trajectories, like how cell is actually differentiating from one cell lineage to another within a given tissue. So how those are actually generated. And then finally, you can you can do a 3D reconstruction of data with the cell level information embedded in that. So these are some of the key applications. And we are not yet able to use all of these possibilities in context of spatial technology either because of the constraints in the computational space or because of the cost. But this is where actually this technology is going to take us to. There are multiple, as I mentioned, like there are multiple ways to make this measurement. So either you have image-based platforms like you have SMFish or Murfish that you are known, the Murfish technology, and the companies like Visgen or the Resolve Biosciences are offering those services to make measurements based on Murfish. Then you have other imaging-based technologies like NanoString, which person talked about recently. Then you have both Genomics and Cosmics platforms, and then 10x Xenium is there. You can also make spatial platforms based on sequencing, like you have 10x Visium or CurioSeeker or CurioTracker is there, or even Stomic SerioSeek is there from Complete Genomics, or you have Singular G4X is there, which is, again, coming from. So all these platforms, they're basically relying on sequencing-based approach to give you a spatial information. Then you have a spatial technologies based on proteomics. So you have platforms like Akoya, you have Luna4, you have Hyperion, IMScope, or Maxima. So this list is not a exhaustive or complete list, because if you go and every single day you will find a new company coming up with a new product. So the only purpose of showing this few of this instrument is to show that how much variety of instruments are available in the market to take advantage of, and then to ask right questions which you want to resolve in the spatial technology. One technology which I would like to specifically introduce you to today here is something called as Atlaxomics. And this is actually kind of a technology which is based on DebitSeek. And I don't know if you are familiar with this, but this is a kind of an open source you don't need an instrument. It's a microfluidic device. You put your tissue and flow your barcode in X, Y, and both the directions. And then that creates a combinatorial barcoding system. And over the top of that, so once the cells are indexed, now you can actually do either expression analysis, attack, seek, cut and tag, or any other thing. So basically, you can build your own technology over this platform. So this is an exciting platform, and we are actually trying to implement that within our core facility. One of the reasons why we think it is interesting, because first thing that it has a very low upfront cost for instrumentation. You only need two things. One, this blue color microfluidic chamber, and then you need a vacuum regulator. So once you have those two things, you can actually essentially do this. And you can actually implement a new protocol, which is not provided by some of these companies. You can actually use this instrument to develop that. So it's as simple as that. You just fix the chip, and then you close your microfluidic chamber, and then you load your barcode, and then apply vacuum, and then again go and load from the other direction your barcode, and then again apply the vacuum, and then just image that barcode positions on a microscope, and then just make ingest library out of that system. So it's a flexible system which not many people are aware of, so that's why I thought that it's good to introduce you to this platform. And so as you saw, like, there are multiple kinds of platforms available. Then what are the major points you should consider when you are starting a core facility, you are starting a service for spatial technology in a core? So one of the major points that you should think about is the instrumentation cost and service contracts. And we will be dwelling on those points more detail, but you should realize that these are mid-throughput instruments, and service contracts will play a big role if you are a cost recovery-based core facility. The other interesting thing, people are when looking at single cell data, single cell resolution, the ability to segment your tissue. Then sparsity, how sparse actually in terms of gene detection. So that is another important thing. And I think during the rest of our discourse, people are going to deal with these points in more detail. Then assay cost, then how you assess tissue quality. This is, again, a big question that you have to think about. Most of the core facilities that are implementing spatial technologies are genomics core. And we are not a histology people. Most of us are not trained in histology. So how we are going to handle tissue quality assessment, and even handling tissue, how we are going to deal with those kind of an issue. This is something that you should think about. Then tissue protocols. Some tissues are easy to handle, like brain. You put on any spatial transcriptome platform, brain will perform fantastic. But when you think about lungs or maybe liver, it's sometimes you need extra optimization. So that is the most important question that you have to ask, like, do you have a different kind of a protocol, a different approach for some of this tissue, and what kind of tissues people are going to use it more often in your core facility compared to others? Then, of course, non-model organism, like Yale-like environment where we are heavily used by human and mouse group, but there are substantial number of people who are using non-model organisms. So how to cater their needs? Do they have something in this party, or they have to just sit and see everybody else is dancing? So this is a question that you have to ask, like, is there something that we can offer? Then again, the same thing, like how human versus mouse versus rest of the species, how we are actually going to deal with that disparity. And then other big question is custom versus off-the-shelf panels. Many technologies are using panel-based approaches where you have fewer panels and then smaller panels. But every time when you talk to a PI, they will say, oh, my favorite gene is not there in this panel. So how you will deal with that? How actually you will deal with the panel? And then not just about the gene, but it's the size of the reaction, the panel reactions. Like in nanostring, you have an incredibly high number of reactions. They offer like 192 with a very, very high cost compared to a smaller package that some of the other companies provide. Then the key question you have to ask that, do you have a strategy for secondary analysis? Because these data it's not microscopy data, where in microscopy you acquire an image, you play a little bit with image or something, and it's ready for publication. Even you are not acquiring the best images, but it's ready for, for your slide, for your publication. So for these data sets, you need a bioinformatician who has to go through this data, create those plots, graphs, everything. So without them, this is not going to happen. So do you have a strategy for secondary analysis? And then how are you going to visualize your data? So these are some of the key challenges that you have to think before you start implementing a spatial transcriptomic platforms within your system. Then the one more consideration that you should think about, that every spatial platforms, they will give you a different QC parameters. But those QC parameters are not equivalent. The meaning of those words seems to be innocently same, but they are not. Like one of the example is FDR. FDR generally calculated through this formula, but if you look at the way Xenium calculates FDR, is actually, it also have number of genes in the panel and then divided by the number of negative controls. So these two formulas looks like innocently similar, but the way that the statistics is being treated is slightly different. So we have to be really, really careful about how we are dealing with these numbers and they all are not. So we have to do some kind of a benchmarking to understand how this is happening. But before I just end, so I'm from Yale Center for Genome Analysis. We are offering multiple services, including spatial technologies. This is our new addition. So we have multiple platforms to offer services from. So I will just leave you with this last question that, I'm from Yale, does it mean that we have access to money, or is it why we need a multiple platform in a given space? So the answer to that is, as I said, like we are a diverse community. And in most of the spatial technology platform, there is a cult following for technology. So it's basically those who are nanostring people, they don't want anything else like nanostring. And you ask them the reason, then they will say, other person was using this, or somebody told them, or they think this is the best. So this kind of a cult following, then we need to have multiple platforms to cater the need of the broad community that we are serving. And of course, we also needed some sequencing-based platform for the reason why I mentioned that. There's a huge community in Yale which are working with non-model organisms. And some people are working as like squirrels and those kind of organisms, and they want to actually use some of this technology as that. So that's why I think we actually went for the multiple. And these are very low throughput instruments, like hardly you can do less than 100 to 200 samples a year. So that means like you can use the AUT for each of these instruments effectively, even you have multiple platform in the core facility. So this is why we went for the multiple platforms. And with that, I'm just kind of handing over to Eric. So this is my group, and these two smiling people, they are my spatial technology specialists. Thank you so much.
All right. Hi, everybody. My name is Eric Chow, and I'm with the UCSF Center for Advanced Technology. And we do a lot of genomics works on campus, but our main mission is to bring new technologies and capabilities to campus. And we've really started in the year 2000, bringing microarrays and use libraries to campus. And again, as the genomics field has evolved, so has our facility. And today, it's really focused on next-generation sequencing and single-cell sequencing with mainly Illumina platforms, but we also recently onboarded a PacBio Revio system for long-read sequencing. We also hold workshops and train users and provide a lot of equipment to users to come in and prepare their own samples. So the majority of the sequencing we do is from user-prepared samples. We do have some people who send samples over to cores for prep, but most of our users provide their own libraries, and so we provide the equipment necessary to do that work. One thing that might be different from us versus other cores is that we're financially independent from the university, which means we get zero support. So there's no subsidy. We don't get X amount of money every year to buy equipment, so we really have to self-fund the equipment and operate under a 100% full cost recovery model. And so this is a little bit different when you think about the economics of bringing on new platforms, because these are investments that we have to get a return on so we can pay for the next instrument in the future to stay competitive. And so that factors into some of our decisions, and I'll walk you through some of that today in this presentation. So just an overview of our capabilities. Again, we're really sequencing focused, and so we have a lot of sequencing platforms. We do have 3 benchtop MySeq systems that, again, are user operated. We don't operate those, we provide training, and people can come in 24/7, 365 days a year to use those systems. And same goes with all the library prep systems, as well as the automation that we actually provide training, and then people come in and do their own programming for their higher throughput experiments. And in terms of some of the requirements for doing spatial work, I think Bony touched upon this, is that we're usually genomics facilities. We don't do imaging, we don't do histology, but we are co-located with an imaging core, and so there would be this opportunity that we could leverage to work with them to bring in some of these spatial platforms. But we haven't done that yet, and so a big reason why for us, again, comes down to the economics, where we have to recover the full cost, including instrument cost. And so what I'm showing here is just like a table, some of the math that I was going through. These are just list prices for all the systems. I'm using the NovaSeq X as an example, and then like a hypothetical spatial platform that has an output of maybe 4 sections a week or 2 sections a week, depending on what you're looking at. And if you factor in your capital equipment, CapEx cost as well, and for that you can calculate your depreciation, say over 3 years, you can change it to 4 years. And then your service contracts on the instrument, if you assume like a 10% instrument cost per year for the service contract, and then an FTE. And if you factor in how many samples a year you could run on the platforms, and take a look at how much added cost you have to put on top of, say, the reagent cost to make up for it, you can see that if you walk through the math, compared with sequencing, we might be adding, say, a little over $200 in these FTE, CapEx, service contract costs per sample, which isn't too bad, considering the cost of those reagents. But depending on your throughput for your spatial platform, this could add quite a bit of cost on top of the reagents that are also fairly expensive. And so here are some of the assumptions: 70% utilization for the NovaSeq. We're actually at higher utilization than that. In some weeks, we're actually at 10 flow cells a week. So for us, this is kind of maybe an overestimate, especially because we can have 1 FTE handle multiple sequencing instruments. Same could probably apply for the FTE for the spatial platforms as well, because they do take a few days to run. So technicians not doing everything on those days. So your FTE cost might be lower, but other costs like your service contract and CapEx cost from depreciation still adds up. And so another way to plot this is just as a chart breaking down these different costs over here. And I think this really highlights, again, depending on how many samples you can run a year, really affects the how much added cost you have to provide the service if you have to recover all of those expenses. And so obviously, if these platforms have higher throughput, these costs would go down. And so again, these kind of high sunk costs and low throughput are really driving these extra expenses that have made it really difficult for us to onboard this, again, given our economic situation. For other groups that can receive funding to buy equipment from the university, again, this isn't as much of a concern, but just kind of the our economics. So for us, again, it's difficult to adopt an in situ spatial platform mainly because of the throughput. I think there's no question the data generated is really amazing and really cool and super informative and really, really useful. It's just, for us, the cost challenge. And then the other issue right now, NanoString is addressing this with kind of larger panels and go into the potentially whole transcriptome. But for a lot of our users, they also right now are new to spatial, and a lot of them don't necessarily know what type of targets they want to go after. So it's hard to figure out a panel for that. And then some of the custom panels are really costly once they figure something, the targets they need. If they're not met by existing off-the-shelf panels, that can add costs as well. And so because of the economics and without a clear user requirement, it's really hard for us to invest in a platform. So this is my last slide, but our current spatial plans are that until we kind of get a sense of what are the spatial requirements that our users really need, we are going to continue pushing users towards NGS spatial platforms. And so Bony talked a couple of these, again, things like Visium and Visium HD, CurioSeeker and CurioTrekker. We think Trekker is also known as slide tags. It came out of the Microscope lab as well, is kind of interesting in that you get single cell data with a layer of spatial information on top. It's going to be interesting to see, a question we have is how much spatial resolution do people need? Do they care if a cell is here, here, or here, or here? I think it's a question that we don't know yet. But if they don't need super high spatial resolution, but they really want whole transcriptome resolution, this might be something that works really well. And then some of the other options that we're working on with some folks are kind of Open and Nova ST. These are like the CScope versions where you repurpose Illumina flow cells to use as a spatial platform. So we're also not ruling out the possibility of bringing in in situ imaging-based platform, but we are definitely thinking about what's going to happen in the field. Again, there's been a lot of things like unknown outcomes from legal issues in the field that have left the future a little cloudy for us and makes it difficult to make decisions. But there are also some new platforms, again, things like Singular and Element both announced different platforms that potentially might be higher throughput. Singular is definitely a spatial platform now, but I think we still need to learn more about the specifications, and I think they've released some more information this morning in a session I wasn't able to attend. And then Element has a TDON system. It's not a spatial platform, but it is an imaging platform, so imagine there might be some type of spatial aspect to that in the future. So we're going to monitor developments in the field and see how throughput changes maybe on new platforms or new assays from existing companies, and then maybe make some decisions later on. And this is just to acknowledge the team. We've grown a little bit over the past year, and so they do really great work and then do a lot of R&D work on the side with collaborations with groups on campus. So thanks for your time.
All right, so my section is going to be very, very small because I'm only here to add a small, different flavor to this discussion. I have great panelists. I don't need to do a whole lot of work. My name is Anoja Perera, and I'm at the Stowers Institute. I oversee a sequencing group. What's very unique about our institute is that we work with all kinds of standard organisms as well as weird critters. It varies from ocean animals to cave crawlers, you name it, we do it. And so we have several things that we need to consider when we are bringing in these new spatial platforms or even any NGS work that we do. Again, it has to be suitable for multiple different organisms. The kit size matter for us. Sometimes we're just looking at a eyeball of a Drosophila, so we don't have a huge need for large capture areas. Resolution obviously matters to us. We have labs that are studying the intestine, and they care about the cells in the intestinal duct. And obviously, like everybody else, the capital costs, analysis pipelines, expertise required, all those things matter. My sequencing group is just like your normal group. The only thing I would point out here is we also have Singular G4 instrument and the Element Aviti. When we were buying those new sequences, we considered the spatial aspect of those. Our journey with spatial started a long time ago, before that time called COVID. We had a really great collaboration with Fei Chen, who's at the Broad Institute. He developed the method SlideSeq, And he used to send us the pucks to do that, do the work. And it was fantastic, and we went on for a couple of years doing that. And then we were super happy when Curio started offering it as a product. But then, all these other new technologies came to the market. And at Stowers, we are not dependent on raising our own funds like the struggles that Eric has. We are privately funded. And we also have like a budget to sort of do some development work. So what we did was we worked with the PI labs and outsourced for like different vendors. We looked at Resolve, Vision. So sometimes, we will work with the vendor itself. Sometimes we'll use their TAP programs. Or sometimes we would even like reach out to our colleagues who have these instruments and say, hey, can you run a slide for us? So Ioannis helped me out with Vision. Mike Hines helped me out with Cinium. And as you can see, like these things took a long time to sort of try out, do the data analysis, because a lot of the data analysis was still being worked out. And also we were trying to like develop expertise in our groups too during all this. And end of last year, we decided to invest in Cinium because we sort of liked that we could throw any organism on it. And we have also worked with 10x for a very long time, so we had great support from them. And then what we have been doing since is we have been ordering assays to do multiple different projects. And we are also looking at maybe bringing in Ph.D. So we are prepared for that. And we are really, really excited about Curio Tracker from Curio Biosciences. So we plan to do like a cross-comparison between Ph.D. and Curio Tracker to understand, and Cinium obviously, to understand which projects can benefit from the different things. So the lessons that we learned during this whole process is spatial is damn expensive. So if you can try out systems before you like invest in something and find something that works for you, that'd be great. And obviously, it should fit your research needs. Like if you're doing just human mouse, you have hundreds of samples to run, like something like NanoString will be perfect. But if you're not, then you should consider other things. And something that I don't think any of us touch on yet is maybe buying is not the best thing. Consider outsourcing, even among us, there are places that will take samples in. There are vendors that have outsourcing set up that you can just send samples to them and they'll process for you. So really keep that in mind. And the field is changing so fast, so think about like if you want to be stuck with something for a long time. Somebody asked me for this recently, so I thought I'll just throw it in there. At Stowers, our workflow looked something like this. Before we do any spatial work, we have a meeting, and that involves a PI lab, the histology, microscopy, and a sequencing group, and the bioinformatics team. So we have a representation from like every group. We discuss the actual project, the spatial options that we can provide, timelines, cost, all of that. And then before we touch any tissue or think about running any sample, we tell the PI lab to go and work with the histology group to optimize their tissue sectioning. Because we don't want to schedule, we don't want to take on something that's just garbage, because that's a waste of everybody's time and money. And then when histology is very happy with working with the PI lab, then they'll schedule time with us, and then we'll do the actual project. And we make sure to capture every single information we can from different groups into our LIMS. So like one struggle we had was, each group worked very differently. Our histology team had a whole different way of capturing information compared to how we capture things. So it was very important, from the beginning to say, okay, we're going to document these things and capture it in a central location so we can, troubleshoot if needed. So once the data is generated, obviously the analysis takes place, and then again, we all come together as a group to see how everything went, if the research questions were answered, if the data quality is good, and that's sort of like the wrap-up of the project. It takes a whole village to do this kind of work. We are so lucky that our histology group is within the same building as us, and we are not that big. We are 25 PIs, but again, we have so many people involved. I just want to highlight Katie, who gave the talk today. And Kate over here. They are my superstars. They do all the heavy lifting. They make me look good. Yeah, so thank you.
Hello. Just a second to switch. All right. Okay, I'll have my timer here. If I go over 10, just throw me something. First of all, very happy to be here. If you were in the morning, this is completely complementary. I mentioned the term production multiple times. Today we're going to touch upon that. I would like to thank the ABRF and the chair for organizing this. And we're the last course standing, so I think last session. And yeah, I'm Ioannis. I'm an associate professor of pathology at Harvard Medical School. I'm also director of the Spatial Technologies unit. The unit, I showed this in the morning, but I'm going to discuss different things. We were established 4 years ago as a only spatial and single-cell facility and with a quadruple mandate to provide democratized access to technology, training and education. And as I said in the morning, that's quite wide. We train from graduate students to physicians to venture capitalists, investors, pharma, what spatial is, how it affects thinking, planning, investing. We support IP, generating new IP, spin-offs, spin-outs, and we're also, I didn't touch upon this at all, but we're also moving technologies to clinical care. Access technology is, which is the service provision that we all majorly do. The focus of today, I won't go in details to this, but two, three major things is we focus a lot on supporting everybody to a pilot, which is great, but we also invest extensively in supporting clinical trials or consortium works or industry clients, which needs a different kind of setup, and I will touch upon this today. And we're all encompassing, we started collaboratively, but we decided in order to control the quality of the work and the timeline, we have to internalize everything. And we have internalized from tissue sectioning, consulting, tissue sectioning, imaging, data analysis, and we go from tissues on our desk to figure 1 to 3 without the project leaving the space. And we provide access to technology. We have this slide, and we provide access technology across the resolution axis. And this is very commonly discussed. What are your major criteria? How come you have these specific things? We're also like Eric, pure cost recovery. We pay for absolutely everything ourselves. So how do you decide how to select equipment? And you'll see very often something like this, plexity and resolution, find the solutions that match your plexity and resolution needs. And you will see a plot like this that has different assays, and you say, okay, I want this one that's up at top right or bottom left. The reality for a core, a lab and a core are different things. The reality for a core is this one. Cost per run, how much real estate you have to work with, throughput. Eric showed very nicely, your cost recovery is bound to your throughput. We work a lot with FFPE, Is it really FFPE compatible? I can throw something 20 years old and work, or it has to be FFPE that was done last night, and we do it in the morning. Data storage, data analysis, how mature it is, the robustness of the assay. You rerun it, or you do a clinical trial with 50 patients or 150 patients. Will it work the same every single time? So other criteria that are very, very important, we're not in a buying spree. Let's buy everything. I wish we could, but we pay for everything, unless NanoString would like to give us stuff. And any other vendor in the room, we accept everything. So, but first of all, is there a need? Is it because I really like it, or actually people are going to use it? Sometimes you do what we call strategic placement, even if not many users need it, and it's an underserved thing, and we need to have it, we might, but usually it's based on cost recovery. Is this need supported completely or partially? Do I have something that I can use this to get similar amount of information, or I cannot do it? If I bring it on, what's the impact? Will it enable people? Will their grants, start coming in? Will people publish on this, or just be happy and have a nice slide to show? Short-term, mid-term investment. There's a star. I'm going to show you how we calculate it. Throughput. 50 times I'll say this. Eric also mentioned it. Indirect return of investment, which is grants and indirects and other things. And very important for us, we always assess this. How is the workflow? Even if the company doesn't support it, can I automate this? Can I make it production grade? If it looks very messy to begin with, probably that might not be difficult for our facility. What's the launch investment? Buy a box with 40K, it's not 40K. Everybody thinks I have 40K, I'll buy a box, it's not 400K, sorry. Service contracts, personnel, just for the launch. You need a couple of months to set up shop, train, SOPing. If the cost per run is for 2 samples, 8K, how many samples you need to run until you bring it, as a line item in your service? And then how many do you need to burn until you fill its production? Will you ever fill its production? Computational biology, because we do the analysis as well. How much time do I need until I can analyze people's data in a fast fashion, not trying to figure things out by myself? Computation infrastructure, if it's generating 4 terabytes per run, for instance, then what do I do about this? Outreach activities, I brought it in. I need to advertise this. I have it. So I'll have workshops. And these are time for my staff. It's a very big investment. And then you expect to have loss in your first 20, 30, 40, 50 runs. I don't know how many depending on the instrument. Who's going to pay for that? Probably you're going to pay for that. So it's not for 100K. It's much, 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 much more. So you have to take this into account. So how do I go from research grade? I bought it. I spent all these hundreds of thousands, and I'm research grade. How do I bring this into production? That's, what is production? High repeatability, low variability, low repeat rate, high volume, high throughput. Like putting the instrument 100% of its workload and with a minimum amount of effort, high efficiency, and low variability. How do you do it? I mean, we use OpenTrons and Parhelia for some aspects. There are other robotic systems out there. We use the BondRx, which is ridiculously expensive, like if you're listening. We use the Next Genomics Connect for single-cell work. These are not small investments. The last 2 are 300K each. So, and then these are just the instruments to enable you to standardize the process, and then standardizing reagents, the workflow, the training, the people. These are multi-$100,000 things. Tissue expertise, we accept everything, like absolutely everything. We do a lot of human, we do a lot of mouse, but we also do porcine models, we do naked mole rat, we do everything that people have. But even for human, bone to brain, they start with a B, but one is the easiest, one is the hardest, and there are a gazillion tissues in between. And then protocol stacking, workflow, okay, and now you have it production, but it's disrupting all other workflow because you need 2-3 people to maximize this. How can you turn around your whole workflow and facility in order to keep it running while other people are doing walk-ins? So that's not an easy equation to do. Major hurdles, lack of best practice, we're making best practice as we go in this field. So it's thanks to everybody's hard work that we have some best practice for now. The protocols of the vendors as they're provided, then we thank them for setting up, at least a start. They're not the protocols we use majorly for almost all our instruments. That small difference in optimizations you do from tissue to tissue, it's money. It's effort, it's time, it's thought, it's exchanging opinions. Turnkey automated workflows, there don't exist. We saw in the morning, that I've seen slide decks now about robotics being advertised for preps, which is awesome. Cost is a major hurdle. I could be testing all day, but if it's 8K per day, then I would close in a month. Sometimes, minimal support. What does minimal support? Many companies do not have the expertise to actually work with complicated tissues. They don't have, histologists in the house. They don't have that level of experience for the complicated things we're doing. And the final things, some instruments are very early. We have, and we're very happy about it, we have many instruments that we purchased them before they even became like a known thing. But you know that there will be work, especially more work, if the instrument is very new, and you'll have to work hand in hand with the vendor and so forth. So am I alone? No, you're not alone. And if you look around, all of these wonderful people are the community we rely on. I wanted to thank Anoja for the WhatsApp. If you're not on the WhatsApp, get on the WhatsApp. We put together the Global Alliance for Spatial Technologies, which is just 1,000 people, 37 countries, I think, only spatial, just spatial all day, protocols all day. Not the same thing, but very complementary. Highly recommend both. Would like to thank, the people that do the science, and I do the slides. And that was my last slide also in the morning. So only 2 slide overlap, if you could allow me. We're always very collaborative. If you're interested in the field, we're always hiring. If you know anybody interested, I won't say no. And thank you.
So we can open up for questions, comments, discussion.
Yeah, Anoja actually threw something at me. I didn't see it.
You were looking away.
You on time, I just wanted to throw something
It should
at you.
be heavier.
So Anoja, I know nothing about this other than what I've just learned. There's been a lot of talk about DataWorks and how we preserve data, present data, make it useful to the end reader. How in the world do you report the results of these spatial experiments in a way that I, as a reader of science or nature or whatever, will be able to see and understand?
I think all of us can answer, but I would say the scientific community sometimes get it. I think there's a bigger issue in getting information out sometimes to just the general public. At Stowers, we have a communications team to do that work. Yeah.
It's not like reporting
Yeah.
a sequence. It's not like reporting a set of SNPs or an association with a gene. It's this gene is next to this gene in this situation with these guys under this type of analysis. Eric's result might be different from Ioannis's result.
100%. So that's a great question. I had only one bullet that we like standards, but what does that mean? I'm a big standard buff, so I believe in logistics. I believe that you need to have the tools in order to do good science. So currently, spatial tissue profiling does not have standards on how to report how an instrument performs. So we rely on the companies making up these specs, but they might mean differently, as Bony mentioned. We don't have standards on how to assess if a run was good today on that instrument. So when you reach out to the vendor and say, I think that run was not really good, then you rely on the vendor, say, no, it looks good to me. Then we don't have standards on how to report our findings in the manuscript, how it should be phrased, in what way, and in what language. We don't have standards on how supplementary data and the raw data should be uploaded to GEO, dbGaP, or whatnot. We don't have standards on how grant mechanisms should be evaluating your pilot data on how to do this. So again, the ABRF, with the groups it has, and I also mentioned Gestalt, we actually have standards groups, people sitting down, and we're working with NIST, we're working with FDA, we're working with multiple to put these things together. But we don't have them, and now it's the good groups and their amazing colleagues out there, they're driving it with putting a good publication out, but we cannot rely that every single person will have the expertise to do it the right way. We have to provide a framework. That's an amazing question. Yes, we're not there.
Yeah.
We should get there tomorrow. In single cell and genomics, it took a decade to get in that level. With spatial, we know what it took in the other fields. So I think we can do a shortcut and not wait 10 years. But we have to all work together.
Yeah, and I would add to that, I would say that because we are working with so many different tissue types, every time it seems like a learning experience. And also because so many teams are involved, it's very difficult to, when something goes wrong even, it's very difficult to understand, where exactly that's happening. And then validation, we can do a spatial, we had to think about, okay, how do you validate the results, right? So we are still working out those things.
So one of the other key aspects is the primary analysis. So each vendors, they are giving a software to us, and then they're extracting information out of an image. So we really don't know that how much equivalent those two extraction processes. So after that, you have a count data, which you can take into a open source software, and you can actually reanalyze it, and you can make sense out of it. But then if anything goes wrong in that primary analysis thing, we have no control over that. So basically, for the investigator's point of view, the reproducibility starts with the primary extraction of data from the image. And at this point, we have no control, neither we have control, nor we have a way to know that what's really happening. And other thing is like those genes which, if you think about the dynamic range of expression, those genes which are expressed moderately, those genes should behave almost similar way in most of the platforms. But especially those which are at lowly expressed gene or very highly expressed gene, they may actually do finicky things in under different primary, the softwares, the way they interact with those data sets. So we really need to have a unified code for those kind of data extraction, which is not available at this point. So that is the first thing. The second thing at Yale, what we are doing really is that we are saving as much data as possible, like as much raw data as possible, because we really don't know that and that is what Ioannis was saying is right, like we don't know what to upload. So we are not giving out anything, and we are not saying like, okay, we only want this particular format, rather than we are just doing bottom fishing. Like you just put a net and fish everything and put as much data as possible into a folder and say like, okay, I'm going to save it for future when I know what to do with this. So I think that is the only possible approach at this point in terms of data reproducibility. And for validation part, we are suggesting now everybody that if you are you are actually basing your interpretation on a certain number of genes. Use some protein-based approach to validate those interpretations what's coming from RNA. So at least there are two methods which are confirming same information, then it's reliable. Other than if you're relying just on the informatics and numbers, then probably I think we are giving too much credits to those approaches.
Yeah, I think you just threw a match, that was not a simple question. Other things I can reveal is with Luciano Martellotto from University of Adelaide and Jasmine Plummer from St. Jude, by the end of the month, we will have at least a first version of clear guidelines tools, for people to use in order to address these questions. Will this be the final? No, it's an iterative process, but at least it will be a compass for those who just enter the field or are trying to feel their way around, but we have to work together.
The cost will prohibit or make it very difficult to do replication on different systems. So your lab gets one result, have Eric do it, or, I'm sorry,
Bony.
Bony do it. I can see how that would be very, very hard to do. And to convince the PI to pay for it.
No, we can.
Can you?
So we are doing a publication with multiple systems, same blocks,
Yeah.
so that's one thing. But through consortia like ABRF, we did, I don't know if you were in
You're
the...
talking about the research group, ABRF.
Yeah, so we did for sequencing, for instance, with the sequencing, we tested different labs, different sequencers.
Right.
Consortia that spanned like with thousands of groups, we could potentially do a multi-group, multi-continent thing if we wanted to, and that's unique. And you don't break the bank because it's a different thing for me to run a few samples instead of running 50 replicates, but we can crowdsource this.
I'm new to spatial biology. We are thinking about one of the first one for our course. Which one do you guys?
There's your second match.
I mean, you have to match I had this slide. Unfortunately, there is no killer application. If there was, we would all have that killer application, and that would be the end of the day. You have to match your users' needs with the right setup. So what do they have? What will they come for you? And then, unfortunately, if you only have a hammer, then you will have to force to squeeze everybody's research through that narrow pathway. So if you find, in general, we try to push people, if they're going to start spatial, start with something where the analysis is easy and you don't go super deep. And also the upfront costs are relatively moderate, so you could enable many people to start. Test the water. If it's like the best thing that ever happened in your institution, then yes, continue pushing this. Otherwise, very kind of very very grim. Like, I don't want to end up with a 350K paperweight.
So you need to see what your users need, right? That's always the critical aspect, and each institution is going to have slightly different needs, so it's really getting in touch with the user base. But if spatial is something totally new, then maybe some of these sequencing approaches that don't rely on an instrument gives people a flavor of spatial and maybe helps them determine what are their actual needs downstream. Do they need more resolution? What are the genes they're interested in? Which companies offer panels that cover those genes, and that might help you figure out which platform is going to be the best fit for your group.
So you're asking us to send out a survey and get...
No, I mean, so...
Talk to people. Surveys not, I mean, we do surveys, but talking to people, but they don't often know what they need.
My take is slightly different on this. So basically, see, if you think about spatial technology, there are 4 or 5 independent components to this technology. One is histology. The second is handling slides. So somebody cutting the slides and then taking this slide and doing something with it. And the third thing is, of course, whatever you image or sequence or whatever you do. And the fourth component is data handling, like how you actually analyze. So the way I think, probably if you are new to spatial, you need to actually work out on all of this logistics. And if you are especially a genomics core who are trying to implement spatial, that means you need somebody who could do the first part of it, like handling slide, cutting slides. So are you going to rely on histology? And then how you are going to get those slides from histology to your core facility, how you are going to store them. Do you have a way to do H&E staining, or is histology is doing that? How you know how you will select FOVs? So all these questions are there, which actually needs to be answered in this order. So my thought was that it's actually, maybe I'm actually wrong in this, but I think one of the best approaches that go for those technologies where you don't have to buy an instrument, and that will give you, like Curio or for Atlaxomics, get hold of this part of the aspect of your operational workflow, like dealing with data, dealing with histology, then training somebody to handle slide. So once you are done and you have run enough number of samples, then you now start implementing new approaches where you have to spend money. And by that time, most probably you already have made a base for, investigators' base for spatial users' base. And they might also have confidence in you that you can do that. So this is, I think, start with a non-capital expense method. And then from there, train your group, align your workflows, create SOPs, and then from there, come back, build your user base, and then come back, and then bring a 450K instrument in the core, and that will be actually a easy transition.
Yeah, so that we don't, you find us next year and say, bad things about us. We don't know your institution. So maybe all the answers we provided are wrong, and the right answer for your institution is get 10 Xenom, 10 Cosmics, and 20 of something else, because you have that need. We do not know. But as Bony mentioned, spatial, either you need to have a lot of buy-in. That's not an easy thing from multiple stakeholders, your histology, everybody. How are you going to move the data around? Or you have one extreme which is completely federated, and the other is our extreme where it's everything internalized. None of this is easy to do. So, and your data quality depends on this perfect synergy of all these parts. If your tissue work doesn't work well, even if your sequencing is perfect, even if your library prep is perfect, you will not get good results. So
I have a quick histology. After that, I'm still thinking the next step is my question. So that's the reason I'm asking.
Yeah, and what Bony said earlier about setting up your pipeline, I think that's very critical. And for us, it really had to work with FAIR because we were getting those pucks for free. So we were able to set that up. One thing for you to consider also is what tissue you're working with.
Cancer tissue mostly.
Sorry?
Cancer tissue.
Okay, yeah.
But cancer tissue can be from bone marrow to kidney to sarcomas. It's a wide spectrum, and for some, the protocols are difficult even for expert groups. Yep. So yeah, find your easy wins. Identify what would enable more funding and grants to come in your institution. Happy to chat afterwards, very specific. But I think it helped also the group see how we think about technologies. Yeah, go.
So, Bonnie, you said something interesting, which you said, I have this menagerie of instruments because I
So for me, actually, I like to have a large panel. And the reason why I'm just working on my ISP rates. And because of the small panel spread across and then multiple options like 2-slide, 4-slide option, I have 25 new items to add into my ISP. And I don't know whether my business office is going to like it or not. So if I have just one big panel, say 5,000 gene panel, 6,000 gene panel, I have only one offering. Then I say, for Xenium, I have one offering, and this is one cost. And if I have, for Cosmics, I have one offering. It's easy for core operations to function. And the other thing is like small panels, that means you have to really buy, store, and you don't know how many users will be using this. So it's kind of a difficult thing from the core operational perspective, having smaller panel. But as many users, especially like I have seen like clinicians who are interested in using Spatial, they are not interested in discovery panels, right? They are interested in smaller panels. They want to actually confine themselves to a smaller number of genes and then add some more custom genes over the top of it. So when you look at the researchers, like research basic science, they want larger panels. So now it's kind of for the core operations side, I think Spatial is going to give us more challenges than we think, or we anticipated from the operational standpoint.
Yeah, I'm very much against Bony's method of doing it because I don't want to be responsible for the PI placing an instrument in my core and having to support that. So one of the things that we did was we evaluated different instruments, see which one works for our institute, and then sort of like justify that process. And if somebody really needs to do like 100 samples with NanoString, we'll just outsource it. We cannot staff it, we cannot spend all that money getting things that 1 or 2 PIs will use.
I also have a strong opinion about that. I'm Team Anoja, probably that's why I'm sitting here. So we're not a supermarket. You can show up and say, I would like 100 of this, like 50 pounds of something. But our goal is for you to be successful. We really, really want you to get the results you need to have your project to be successful. And all these nuances with the different technologies and how the data would look like and how the specific cell populations you're interested in will look like, it's almost impossible for somebody not working behind the scenes know these things. So you can come we do a consultation. In the consultation, we will ask specifically your research questions. We will write these down. And then we will match your research questions with one technology, with multiple technologies, and how 2 or 3 scenarios that are potentially possible or more, how much they will cost, how much time they will take, and what data you will get, and how your questions will be answered. Of course, at the end of the day, it's your project, and you might say, like, to hell with it, I really want to do it with this. But it's our role to provide our expertise and guidance to enable you to do your science, not to just shop something that I will make 5 times more money than something else. Sometimes we tell people you don't need Spatial for this. I'm very sorry. That's a huge project. I would love to have $500,000 of your money, but you do not need Spatial for this.
I know we are over time, and I know we said we won't share our opinions about things, but come find us and we'll tell you all about what we do.
Thank you.
Thank you so much.
Thanks for organizing.
Thank you,
Thanks, Bony.
Thank you, fellow.
Thanks, Katie.
Ioannis, I have a question.